So this is a huge thing that's happening now. But it's all based around this distinction of the drug content in hemp and marijuana. And so onto my research and how that is informing the development of this kind of public policy. So we're going to dive into the genetics of cannabis, and I'm going to refer back to a mentor of mine who was the professor who founded this field of ethnobotany, who happened to study cannabis around the world back in the 1970s, Richard Evans Schultes, and he has the notorious claim to fame of producing the golden guide to hallucinogenic plants. Now, if any of you know the Golden Guide series, it's a great way to get to know your gems and minerals, your birds and butterflies, and so forth. For some reason, the publisher of the Golden Guide enlisted Dr. Schultes to produce a Golden Guide to hallucinogenic plants. Well, as soon as this hit the stands, you can imagine what happened. It's very limited release. Pulled immediately. Very fundamental concepts here is that we have all this variation out there in nature, that some of it we can explain by different genetics, and some of it we can explain by different environments. And that's illustrated very simply if you look, you put, let's take two different breeds, like breeds of dogs, hemp and marijuana. You grow them under the same conditions, like at a low density, and you see that they look different. These marijuana plants look a little more highly branched and shorter than these particular hemp plants. So that's that genetic component, and that's what I've been interested in investigating. There's also this component that we're also interested in, which is environment. If you put these plants into different environments, you also observe differences. We try to tease apart what we can explain by genetics and what we can explain by environment. Nature, nurture. You take hemp plants and you grow them at low density, they're more highly branched than if you grow them very closely planted together, they're less branched. So the particular genetics that I'm interested in is how these cannabinoids, these unique molecules that only cannabis plants produce, how do they vary among different breeds or strains or varieties of cannabis. And I've mentioned THC, the euphoria-inducing molecule that we find in marijuana plants. Well, there's a closely related molecule called cannabidiolic acid, or CBD. And it turns out hemp plants produce mostly CBD. This chemical also has pharmacological effects, but it's not really a euphoric kind of thing. We'll talk about some of the other effects later. But these kind of two major types of cannabis produce either a lot of this or a lot of this. And I've been interested in trying to understand, how is that so? Why is that so? We can think about, we can look at this question, why marijuana produces mostly THC but hemp produces mostly CBD in the context of biochemistry. And it turns out that these two molecules, THC and CBD, are they're alternate products of a common biochemical precursor called cannabidiolic acid, or CBG. If you think about biosynthesis like a factory assembly line, early in the process you have this thing, and then you convert this thing either into that or into that. The chassis in the Ford plant is the same at one step, but then you add a different body onto it, and you make one type or another type of car. So we published a paper last year that gets at the genes that are responsible for either making a lot of THC or a lot of CBD in these plants. It represented about 12 years worth of very slow, painful work. Slow because we were over-regulated, denied the opportunity to do simple experiments. Slow because we were also woefully underfunded. Let me walk you through what we discovered. Requires a little background genetics. So cannabis, just like the human species has 2 copies of every chromosome, and each parent contributes 1 copy, one of their 2 to the offspring. So that's diagrammed here. And these chromosomes are the strands of DNA where genes are located. Those genes are the specific instructions. So what we did was take advantage of the variation that exists between hemp and marijuana in drug content by importing some Dutch marijuana under DEA permit to my lab and some Canadian hemp. We grew those plants up in our minimum security prison facility. And that's really what it is. And that's what I tell my students they're working in. And we produced a hybrid. We crossed these 2 to get a marijuana by hemp hybrid. And then the next thing we did was take that hybrid and we self-pollinated it. We were able to trick the plant into fertilizing itself and producing its own seed. And then we look at the grandchildren. And there are these processes that go on. One is how chromosomes sort out, and the other is how DNA recombines during this process of cell division, where we're then able to look at how does drug content sort out in the grandchildren of this hemp by marijuana cross. I'm sorry that this is the data slide. Try to stay away from data slides, but this is the one data slide in the talk, and it'll recur a couple more times. What is shown here is a plot of the percentage of the CBD and the percentage of the THC. Each little point on this chart represents a single plant. And if you just kind of squint, you'll see there's 3 basic kind of lines. There's plants that have a lot of THC and very little CBD, almost 0. And there are plants that have a lot of CBD and very little THC. In red circles are the marijuana parents, and in red squares are the hemp parents. There's another class here that has a bit of both, and those are the hybrids and some of the grandchildren. We grew up 500 grandchildren, and we found that those grandchildren fell into a very simple pattern. They either looked like the marijuana grandparent, the hemp grandparent, or an intermediate. And the ratio was 1 to 2 to 1. So if any of you can harken back to your high school biology, and you might have learned about this Czech monk named Gregor Mendel who discovered the simple laws of inheritance in 1965, it's the simple pattern. You know, if you take a red snapdragon and you cross it with a white snapdragon, the hybrid might be a purple snapdragon, then you self-fertilize this purple snapdragon, and the grandchildren give you our 1/4 red snapdragon, 1/4 white snapdragon, and 1/2 pink snapdragons. And that's exactly what we saw. So let's imagine that there's some gene on some chromosome where there's a specific region on that gene where there's different variations in the DNA sequence. Maybe a variation that produces THC and a variation that produces CBD. Let's suppose we had that. And imagine that, you know, when the plants make their pollen and they make their ovules, these copies get separated and sorted out. You would, by chance, expect to get 1/4 plants that would have 2 copies of the THC gene, 1/4 would have 2 copies of the CBD gene, and 1/2 would have 1 copy of each. And that would give you this 1 to 2 to 1 basic intermediate pattern. Well, it turns out biochemists have discovered the enzymes in the biosynthetic assembly line that convert the precursor molecule into either THC or CBD. So could it be that these 2 enzymes are proteins are coded by DNA. Could it be that there's a DNA region where these different enzymes are just coded by different variations of DNA in the same gene on the spot in the chromosome. That's the simplest explanation for the pattern that we saw. And so we tested that through DNA sequencing, because we know actually that we know what the genes are for these two enzymes. The one enzyme is called THCA synthase, and the other enzyme is called CBDA synthase, and those proteins are encoded by these specific DNA sequences, and I can tell you that they're not just variants of a single gene, because we find DNA sequences for both of these enzymes in both marijuana plants and in hemp plants. What our work has shown is that instead of just having a single gene, we have two separate genes, at least two, maybe more. One for THC synthase, one for CBD synthase, and they're very closely linked together on the chromosome. DNA sequences can duplicate themselves, and you can get these repeated tandem units, and that's what we think is going on here. It's the only way to explain the pattern that I just showed you of the 1 to 2 to 1 and the presence of multiple copies in this genome. You can see that, let's say in the gene sequence for THCA synthase, there are some differences in the underlying DNA sequence between these types. There's one really surprising difference we came across, and that is that in the marijuana type plants, the gene for CBD synthase has 4 DNA letters deleted. And that turns out to be quite important to explain the difference between hemp and marijuana. We have to go even deeper into molecular biology now, but to understand why this might be so. And it's the idea that, DNA is the information-containing molecule. It gets transcribed into this RNA molecule, then the RNA molecule gets translated into a series of amino acids into protein. The machinery that translates the RNA into a protein depends on the match between three of these nucleic acids and a particular amino acid. When this ribosome, it's called, is chugging along this molecule, adding amino acids, extending the protein chain, when you get a deletion happening, it throws off the sequence of translation. It often results in the sudden termination of protein translation. And so you end up, when you have a deletion like this, you don't produce a functional protein. So we now have a very simple explanation for why hemp and marijuana are different. The hemp has a functional CBDA synthase enzyme. This is what the protein looks like according to biochemists. But in the marijuana strains, there's this deletion and you don't make a functional protein. So hemp isn't marijuana because marijuana lacks the enzyme that makes CBD. Why is that so? Well, because these two enzymes, they compete for the same precursor molecule. And if you knock out one competitor, the other one wins, and you end up with a lot of THC. It's a pretty simple thing. The marijuana plants seem to be lacking the enzyme that makes CBD.